Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we will be continuing to discuss ordination, in this case, constrained ordination. And we're going to use correspondence analysis as an example. So we have talked about correspondence analysis, although not so much directly. So correspondence analysis is basically the same thing as doing chi-square distances on your feature table, like your species table, and then applying principal coordinates to it. Although there's another layer to it that we haven't talked about. So we discussed how the chi-square distances are very good at recovering gradients if there's a strong primary gradient affecting your samples. And it turns out that this is because correspondence analysis as a technique is guaranteed to recover that gradient under certain conditions. So the condition under which it does this is when each species has a unimodal response to some environmental gradient. So basically, let's say for example with the Guerrero-Negro data, if your gradient is the depth of sampling, and so let's put that on the x-axis here. So this is the top of the microbial mat, and this is going deeper into the microbial mat. Each species is assumed to have a peak somewhere along that gradient. So either they're highest at the beginning and then they're monotonically decreasing as you move along, or they're highest at the deepest part and they're monotonically increasing as you move along, or they have a peak somewhere in the middle. So you'll see them increasing and then decreasing. So that's what we mean by unimodal response. And if every species has a unimodal response to some primary gradient, then correspondence analysis will figure out what order the samples need to go in order to best recover those unimodal response curves of the species. And when it does this, it's putting the samples in order, and it's also explicitly putting the species in order, or figuring out where the mode is of that species along that gradient. So you can get these plots that look like the one shown here, so this is actually the Guerrero-Negro data shown again. It's going to look pretty similar to the chi-square plots we made before, except here it's a biplot. So we have the small dots are the samples, and then the red dots are the small black dots are samples. The even smaller red dots are all of the OTUs that we left in the table. And so this technique is used to make a biplot like this commonly. And we're also going to use a constrained version of this that works well when you want to explicitly test for certain gradients. Now, when you do one of these plots, you can ask the same type of question that some people have asked in principal coordinates analysis, where you want to know how much variance is explained in the first axis or the second axis, third axis, and so on. And it's still the case that it's not necessarily relevant. So you might have more gradients in your data, so you can't explain as much on the first axis. Or maybe chi-square isn't as good a distance metric, and a better distance metric would allow you to explain less variation, but recapture something interesting biologically in the data. So the absolute amount of variance explained on each axis still is not highly relevant. I mean, I wouldn't base your publication on whatever fraction of the variance is explained here, but it is important in terms of relative comparisons between unconstrained and constrained ordination. So we're going to see more of that later. When you run correspondence analysis, the easiest way to do this is in the vegan package in R. And we've already used it. If you look at the code browser for the chi-square beta diversity, we actually used correspondence analysis to get the chi-square ordination out. And when you do it, you end up with this object, which I've called my.ca, so it's my correspondence analysis result. And if within that object there are a whole bunch of elements, one of them is the correspondence analysis list, which is basically just a whole bunch of other results from the analysis. So if you use a $sign and then CA, that'll get you into the correspondence analysis part of the results. Then another $sign, EIG. Those are the eigenvalues, which are basically the amount of the total variance that was explained in each of the axes. And then the total variance in the whole data set is the total chi-square distance. So you can access that with $sign tot.chi. And so if you divide the eigenvalues by the total variance, you can see what fraction of variance was recovered. And here in this analysis, we get something, 30, 40% in the first axis, something in the 20s for the second axis. Okay, so that's unconstrained indirect gradient analysis, where we're just ordinating. The method doesn't know anything about depth or pH or any oxygen content, any other geochemical gradients. Now we're going to use the same method, but with direct gradient analysis. So here we're actually going to pass in specific covariates that we think are driving variation in the community structure. And so we're directly trying to relate the species response curves to some covariates that we have collected experimentally. Now, if you read Mike Palmer's page, very nice page about ordination techniques, he describes canonical correspondence analysis, which is also sometimes called constrained correspondence analysis, as a marriage between correspondence analysis and multiple regression. And I'll explain why. The constrained correspondence analysis is also trying to maximize the correlation between the, essentially find the best unimodal gradients for all of the species. And but the difference is in constrained correspondence analysis or canonical correspondence analysis, the positions of the samples along the first axis, second axis, and so on have to be a linear combination of the covariates that you gave to the algorithm. So because of this, the constrained analysis, or CCA, has to explain less variation than the raw unconstrained CA. So the unconstrained CA can put the samples anywhere in order to maximize the amount of the total variance explained. Constrained analysis has to position the samples as a linear combination of the features that you give it. A rough approximation to the algorithm for this is to do correspondence analysis. This gives you the chi-square distances. Then, so we get chi-square distances. We perform correspondence analysis, so that gives us the positions of the samples along the first axis. Then we regress those optimal positions on the covariates that were provided. And what we're left over with are the constrained first axis scores. Then we repeat that for the higher axes. And we're using those fitted coordinates as the scores along axis 1, and then we do it for axis 2, and so on. Okay, so let's see what a constrained correspondence analysis looks like. This is also using the Guerrero-Negro data, and we use the same call to this function from the vegan package, so it's the CCA function. But here we're giving it the OTU table, and we're telling it that the OTU table is varying as a function of these covariates. So end depth, that's the depth end of the sampling of that particular layer. The fraction of chemotaxis genes that were observed in that sample, and then the fraction of flagella that were seen in that sample, and or genes for flagella. And these names here are actually the names of the columns in your mapping file or your metadata table. And the way these functions work, whenever you have a dependent variable, tilde, independent variables, you're allowed to just use the names of the independent variables. And then there's another parameter called data, which where you tell the function what table to look for those variables in. So that's what we're doing here. OTUs varies as a function of depth, chemotaxis, flagella, and the data structure that contains those values is the map variable. So anyway, we call this function. Vegan does the constrained correspondence analysis, and then we can plot that, and this is the plot that we get. So we see that it looks somewhat similar to the unconstrained plot. You can still kind of see a gradient in the black dots, although it's not quite that perfect recovery of the depth gradient that we saw. And you still see that there's some distribution of where those OTUs, the red dots, where they are falling along the x-axis and y-axis. They kind of follow along with those black samples still. And then, so that's our biplot, the black dots and the red dots. But we have a third component, which is the contribution of each of the covariates to the sample scores along each of the axes. So it's basically the regression coefficient, the direction and the magnitude of the regression coefficient. And so let's see what we see here. Depth looks like it's increasing as we move toward the right. Then, so we still basically have a depth axis along the x-axis where we're getting deeper as we move along to the right. Then chemotaxis is increasing up toward the left. So basically, chemotaxis genes are following the opposite direction of the depth of the sample. So the more shallow samples tend to have more chemotaxis genes. And then, interestingly, flagella is somewhat orthogonal to these. So flagella does not seem to be increasing substantially with depth. Maybe it's, it's slightly going in the same direction as depth, but for the most part, it's associated with this other axis, axis 2, whatever that is. So it's basically an orthogonal feature to depth, not highly correlated with depth. Whereas chemotaxis and depth are going to be highly anti-correlated. So that's what we call a triplot. We've got the species in black, the samples in black, the species in red, and the contributions of the environmental variables to the species, the sample scores in the arrows. We are now ready to use the variance explained to compare this to the unconstrained correspondence analysis. So you get the values of the percent variance explained in the same way here, almost the same way. So in the my.cca object, that was the result of the vegan package function call. You use $, here we're going to go into the CCA part of it, then another $, to get out the eigenvalues. Then same thing, we divide those by the total chi-square distances, and that gives us the fraction of the variance explained by axis 1, 2, 3, 4, and so on, which I've shown on the plot here. And what we see is it's a substantially lower amount of variance explained. In this case, we had the actual numbers for the first one were 26.4 for correspondence analysis axis 2 and 36.7 for the first axis. Now we have 31.2 for the first axis, 10% for the second axis. So they've gone down substantially. Also note, I flipped the correspondence analysis axis upside down. Remember that the direction doesn't matter. I just did it so that it would kind of line up with the constrained analysis. So is this a good result? Well, it's potentially informative. It makes a neat little plot, and you can start to tell more of a story about what's going on biologically here. But it's not clear how to test the significance of this. So one very simple thing you can do is just state the fraction of the variance explained state the fraction of the percent variance explained by the unconstrained method that is explained by the constrained method. So 31.2 divided by 36.7, so we're getting about 3/4 of the variance explained by, in other words, you could say, in this data set, these three covariates were able to explain about 75% of the variance of an optimal gradient in the samples. Now, we can also test whether or not constrained correspondence analysis is significant explicitly. And the way that we would do this is we have to think about what the null hypothesis is. So there's a very simple way to do this using what we call Monte Carlo simulation. That just means let's make a bunch of scrambled data sets and keep running the method every time, and we'll see how good those look. Then we compare the actual results to those. So that's Monte Carlo simulation. These can be a little bit tricky. So we saw an example earlier in the course where a simulated data set was actually using probably less valid null distribution for simulating species abundances. And so in that case, it basically inflated the p-value. It made the p-value look a lot better for the actual data. Here we're not trying to simulate microbiome data. We're just reordering the labels of the samples so that, and then we're still using the raw values or the original values for chemotaxis, flagella, and depth. And the distinction here is that we're not trying to figure out what distribution chemotaxis values come from and then simulate a new set of chemotaxis values. We're just rearranging the original values. So we're staying true to the original distributions. Also, there are a couple different ways to do this. You could scramble, well, let me show you an example. This is an example of constrained correspondence analysis using scrambled or shuffled values of chemotaxis, flagella, and the depth. What do you see here? Well, the nice gradient structure is gone, and now we see that all the OTUs are basically a big blob in the center, meaning that they did not get distributed in a nice unimodal way where some of them are closer to one end, some are closer to the other, and some are in the middle. They're basically a giant blob. And you can see also here the depth is hardly correlated with the first axis at all. The way I generated this plot was to simply reorder the samples. So basically keeping the chemotaxis values with the depth values with the flagella values. And you can see here that chemotaxis and depth are still going in opposite directions, and flagella is still orthogonal. So we're controlling for the correlation structure between the covariates by keeping them together and just reordering the rows of the mapping file. We can do this, say, 10,000 times. Then each time we measure what fraction of variance is explained by axis 1. And then we see what fraction we explained with our actual covariate values. And that gives us a p-value. Basically, what percent of the time did the randomly generated percent variance explained look better than or equal to our observed variance explained. And so the Monte Carlo simulation is pretty simple. It's just rerunning the analysis lots and lots of times on scrambled data and comparing the results to the observed data. And then that gives you a p-value that you can report. I want to show one other example of this in use. So I actually used this in a paper a few years ago. This was an analysis with Jeff Werner and Rob Knight and Lars Angenant of the microbial communities at a bunch of bioreactors for treating wastewater at breweries. And what we saw was that the community structure was very unique for each of the different types of reactor. Well, so it was unique for the different individual reactors, and then also somewhat unique for the different reactor types. And one of the funny things about this that came out was, so these guys were at Cornell when we were doing the collaboration, and the Cornell newspaper put out a picture of someone pouring Ithaca beer into a gas tank. So that wasn't exactly what our finding was, but we did find some interesting things out about how certain species might be driving the stability of these bioreactor communities, which might eventually help improve the industrial fitness, like production of biofuel. What I wanted to show you was that this paper actually has a number of examples of doing ordination, unconstrained ordination or constrained ordination. And we explicitly used this comparison where we show correspondence analysis of all of the samples. And in this case, we were able to explain 6.6% of all of the variance in the data set. And here you see the nice clustering of the different bioreactor samples. And then we did constrained correspondence analysis using methanogenic activity, temperature, a few other metrics that we were measuring. And we saw that with just these 4 variables, we were able to explain 5.5% of the variance. So this meant that we were getting whatever it is, around 80% of the variance explained. In other words, indicating that these variables provided nearly the same explanatory power as an optimal gradient. So in summary, CCA does direct gradient analysis. I recommend that you never use more than 3 or 4 variables in this. Once you get up to 5, 6, 7, 8, you're going to start seeing spurious associations unless you have a really big data set. So more than 3, 4, 5 is going to start overfitting the data. You can measure the success of the constrained ordination by the ratio of the variance explained, the constrained variance explained to the unconstrained variance explained. So like in the example we saw, our canonical variance explained about 85% of the total explainable variance in the first axis. And I also want to point out that canonical correspondence analysis is the same as constrained correspondence analysis, so they're both called CCA. But there's another CCA, which is canonical correlation analysis. And that's something completely different that we might talk about toward the end of the class. That's all for this video. See you next time.